Today, I would like to address an issue that affects millions of people worldwide, yet remains largely misunderstood outside of medical and scientific communities: the impact of low blood sugar on eye health, particularly in individuals living with diabetes. For decades, the focus of diabetes management has centered on preventing high blood sugar. While this remains critically important, emerging research now reveals a more complex reality. Dangerously low blood sugar levels, known as hypoglycemia, may also cause serious and lasting harm. Recent scientific findings show that hypoglycemia can directly damage the eyes and may play a significant role in the development and progression of diabetic retinopathy, one of the leading causes of blindness worldwide. Today, I will discuss groundbreaking research funded by the U.S. National Institutes of Health and conducted by scientists at the Wilmer Eye Institute at Johns Hopkins Medicine. This research offers new insights into how low blood sugar affects the eye, why diabetic patients may experience worsening vision even when attempting tight glucose control, and how future treatments may help protect sight. To fully appreciate the importance of this discovery, it is necessary to understand diabetic retinopathy itself. Diabetic retinopathy is a disease caused by damage to the small blood vessels in the retina, the light-sensitive layer of tissue at the back of the eye that enables us to see. Over time, prolonged exposure to abnormal blood sugar levels weakens these vessels, causing them to swell, leak, or grow abnormally. As the disease progresses, it can lead to blurred vision, blind spots, and eventually permanent blindness. Traditionally, high blood sugar has been identified as the primary cause of this damage. As a result, diabetes management strategies have long emphasized strict glucose control. However, the new research challenges the assumption that lower blood sugar is always safer, revealing that episodes of hypoglycemia may also have harmful consequences for the eye. At the center of this discovery is a structure known as the blood-retinal barrier. This barrier functions much like the blood-brain barrier, acting as a highly selective filter that controls what substances can move between the bloodstream and the retina. Under normal conditions, the blood-retinal barrier protects the delicate retinal tissue from toxins, excess fluids, and harmful molecules. It ensures that essential nutrients reach the retina while keeping damaging substances out. When this barrier is compromised, however, fluid leakage and inflammation can occur, leading to vision impairment. The recent study reveals that low blood sugar can weaken this protective barrier, setting off a chain reaction that ultimately damages the retina. Researchers at the Wilmer Eye Institute conducted a series of experiments using both diabetic and non-diabetic mice to understand how low blood sugar affects the eyes. They focused on a protein known as hypoxia-inducible factor, or HIF. HIF is a protein that helps cells respond to low oxygen levels. Under certain conditions, it activates genes that stimulate blood vessel growth and alter cellular metabolism. While this response can be helpful in some situations, excessive or inappropriate activation can be harmful. The researchers discovered that when blood sugar levels dropped, levels of HIF rose significantly in the retinas of diabetic mice. This increase did not occur in the same way in non-diabetic mice, suggesting that diabetes makes the retina particularly vulnerable to hypoglycemic-induced stress. As HIF levels increased, it triggered a cascade of harmful events. The protein activated other molecules that caused abnormal blood vessel growth and increased permeability in the retinal tissue. As a result, the blood-retinal barrier broke down, allowing fluid and proteins to leak into the retina. This leakage is a defining feature of diabetic retinopathy and a major cause of vision loss. This research is significant for several reasons. First, it challenges the long-standing assumption that tighter blood sugar control always leads to better outcomes. While maintaining healthy glucose levels is essential, this study suggests that frequent or severe episodes of hypoglycemia may actually accelerate eye damage in some individuals. Second, the findings help explain a clinical mystery that has puzzled doctors for years. Some patients with diabetes experience worsening retinopathy even when they appear to be managing their blood sugar carefully. This study provides a biological explanation for that phenomenon, highlighting the role of hypoglycemic-induced stress on retinal tissue. Third, it underscores the importance of balance in diabetes management. Avoiding both chronic high blood sugar and dangerous drops in glucose levels is crucial for protecting long-term vision. Perhaps the most promising aspect of this research is the identification of HIF as a potential therapeutic target. The scientists tested a drug known as 32-134D, which blocks the activity of HIF. When diabetic mice were treated with this drug before experiencing low blood sugar, the results were striking. HIF levels remained controlled, the blood-retinal barrier stayed intact, and the damaging cascade that normally leads to retinal injury was effectively prevented. These findings suggest that medications targeting HIF could one day be used to protect the eyes of people with diabetes, particularly those who are prone to hypoglycemic episodes. Such treatments could work alongside existing therapies, offering a new layer of protection against vision loss. For patients living with diabetes, this research sends an important message. Glucose control is not just about lowering numbers, it's about maintaining stability. Frequent swings between high and low blood sugar may be just as harmful as consistently elevated levels. For clinicians, these findings reinforce the need for individualized diabetes management strategies. Treatment plans should consider not only average blood sugar levels, but also variability, hypoglycemic risk, and the patient's overall metabolic health. This research may also influence how clinicians monitor diabetic eye disease. Earlier screening, closer observation of patients with frequent hypoglycemia, and future use of targeted therapies could significantly reduce the risk of vision loss. The researchers involved in this study, led by Dr. Akrit Sodhi, have emphasized that this work represents only the beginning. Further studies are planned to explore how HIF functions in human patients and to evaluate the safety and effectiveness of HIF-blocking drugs in clinical trials. If these therapies prove successful, they could transform the way diabetic retinopathy is prevented and treated. Instead of focusing solely on managing blood sugar levels, physicians may soon be able to protect the retina directly, preserving vision even in high-risk individuals. This research also opens the door to exploring similar mechanisms in other diabetic complications, such as kidney disease or nerve damage, where blood vessel integrity plays a critical role. In conclusion, the discovery that low blood sugar can damage the eyes represents a major advancement in our understanding of diabetic complications. It highlights the delicate balance required in glucose management and underscores the importance of protecting the body's natural barriers, particularly the blood-retinal barrier. The identification of HIF as a key driver of retinal damage offers new hope for patients and clinicians alike. With continued research and innovation, it may soon be possible to prevent vision loss more effectively, improve quality of life for millions of people with diabetes, and redefine how we approach diabetic care. As we move forward, this research reminds us of a vital truth. In medicine, balance matters. Protecting health is not just about controlling numbers, but about understanding the complex biological systems that keep the body functioning as a whole. Thank you.